S9 in application to some non-Drosophila, non-red organisms.
Yeah. Thank you very much. If you allow, I speak English. Is it okay? Yes, so thank you very much to organize. It's my great pleasure to be here. And being on a final track of the conference, it's very difficult to make something original. So, I made a choice, so I give you something exotic and something motivating to action, so it's kind of two parts in my talk, and let us start. So actually I represent this Japanese-Russian laboratory that chose a path to survive in science by focusing on some extreme events in biology. So a laboratory located in Kazan and in Riken are keen on organisms with a very exotic, very unusual capabilities. And I give you some stories about that today. So we have very interesting structure. We're actually a very young laboratory, and we have two sites, one located back there in Kazan University, and the second located back in Japan and Riken. So we exchange by scientists and transfer technology. And recently it became much easier to explain to Japanese colleagues where Kazan is, because the soccer Japanese team been camping in Kazan, so now people are more accustomed. So before we've been mistaken with Kazakhstan, so so Kazan and Kazakhstan is kind of the same place. Right, so as I mentioned, we are focusing on some unusual creatures, and we study very extreme organisms, including chickens, so believe me, it's quite interesting stuff. But today, I'm going to talk about our favorite model, a midge, and technically the bloodworm, which is kind of larva or with midge. Yes, so would you please click? Right. So in appearance, this bloodworm looks like a very usual one, but it technically has very unusual capacity that you can technically desiccate the whole body of this creature, and you can keep it on the dry stage for decades, maybe hundreds of years. And later, with a drop of water, less than 30 minutes, it's come back to life, and it's continuing activity, and you can do it many, many times. So it's a kind of natural, excellent survivor. And one example is this: you place it outside of the space station with the help of the Russian screwed team. Later, it will come back after 2 years, which is kind of fly to Mars and back, and will completely dehydrated and maintain activity, so we completely survive. So it's a very nice guys. So for biologists, for us, it's quite interesting that this capacity to survive without water is actually not limited to the whole organism as a piece, but even parts of this midge, of this larva, can come back to life. If you chop off the head, so you cut off the brain, you can desiccate this guy without brain, and the tail will revive later and maintain living. So what we know, separate tissues or extracting organs can be also preserved in a dry stage. So it's a very interesting model. And what we are lucky about, we have a first and only cell line that you can completely desiccate and keep for years, and a larger part of the cells will actually revive after adding water. So it's a kind of model for biobanking, and you can, it's a kind of illustration that actually cells can be modified to have this capacity. So just to give a long story short, we work a lot with genome science, and two things I want you to remember, but the two secrets in this small creature we know. The first, that it doesn't keep water inside when you desiccate it. It's completely release all water from the body, and it replaces it with the sugar, with the trehalose. Probably female scientists know more about the trehalose because it's a component of the cosmetic cream to maintain moisture. And in this guy, there's actually 97% of the body in dry state are occupied by trehalose, so it's literally completely desiccated. So when it's dry, you should better keep it in vacuum. It will be much happier with under normal humidity. And what we know about after sequencing of the genome of this creature, but it makes quite interesting modification to its own genome in the course of evolution. And one of the quite interesting and exciting part, if you compare this guy with a common ordinary midge with a kind of in the same group, you will see where the part of the genome has expanded to form more paralogous copies of certain genes that are only become active under desiccation stress. So they have very specific, interesting set of genes that specifically involve to cope with the desiccation. Just to give you two examples, so we call these pieces in the genome islands, which is related to this anhydrobiosis, to this capacity to live without water. And we know that there's two major mechanisms how it's evolved. And one is a classical horizontal gene transfer, with an example of quite interesting so-called LIA proteins that are found in a bigger number in the plants and specifically accumulated in the plant seeds that allow plant seeds to be kept under dry stage for thousands of years. Some other enzymes being grabbed from bacteria and also effectively used in its genome. The second way how it happens is just drastic expansion on a certain classical genes. It looks like the genes first parallel jump some other place of the genome and then expanded in number and become specifically activated in response to water. And if just to summarize in one sentence, this guy have an extra set of antioxidants, it has extra set of heat shock proteins, and even I use it for commercial promotion of our project. It's anti-aging enzymes being expanded in the genome. So it's kind of extra set and it's specifically responsive to desiccation only. Just to give you a very simple example, so this creature is actually a midge. Only larvae can survive without water, and it's example of thyroid things that people who work with antioxidants knows about this protein, and the classical available in all insects proteins on RNA and protein level can be detected on every life stage, and those specific guys that only found in this creature can be seen in active only in the larva stage and only under desiccation. So it's a quite interesting and smart decision of evolution. To go closer to the topic of our conference today, our very recent finding has been done in collaboration with Sculptech and recently published in PRONUS. We found that actually there is some logic. So it looks like this very unusual midge has hijacked that own heat shock response system, and we found a lot of motifs that are recognized by heat shock factor in front of the genes that specifically react to desiccation. So these motifs are known from Drosophila as a heat shock elements, as a binding motif, and they found a lot in the front of genes that specifically contribute to desiccation resistance. So just to remind, the heat shock response system based on aligning of monomers of heat shock factor under stress, and then the heat shock response activated once a trimer induced and bounded to heat shock element in the genome. And what we see that actually this heat shock factor and drastically induced specifically in this midge in response to desiccation, not in the usual midge. And once you make RNAi shutting down of this heat shock factor, the survival rate of the cells, you know we have this in vitro model, drastically drops. So it was the first kind of motivating confirmation for us, and we decided to go further into analyzing this function using genome editing that had not been available. So we're actually just looking on the structure of the gene of heat shock factor, again in this single midge. This is our specific midge, and this is kind of its relative that cannot stand desiccation. We see there's a basic shape of the gene, and specifically alternative splicing events is a completely specific in a polypedilon van de Planck, which is our X midge. So interestingly, so the splicing makes several isoforms on the heat shock factor, and depending on the cycle of this desiccation, drying, and then rehydration, the specific isoform 2 is a highly abundant, and it looks like we're hijacking this kind of tricking with the own heat shock factor being realized in the form of making specific variant to be activated in the course of desiccation. So when you work with such strange creature, you always face the situation where you don't have methodology. It's a quite exciting, it's a quite exotic creature, but you always need to think how you will be applying technique. And we were lucky, so one very exciting feature of Diptera cells, and the people who work with Drosophila know it, you can easily make a stable cell line by introduction of a foreign gene. So if you inject the vector containing some protein of your interest with a proper promoter, these Diptera Drosophila cells integrate them in their own genome, and you have kind of randomly integrated in the genome kind of stable line. So what we did, and we had specifically GFP expressing PB11 cells, these cells I've been talking about before. And what we did, we first just try a very simple experiment by co-transfection of heat shock factor with specific isoform we've been interested in, and with heat shock elements containing motifs. And specifically we see that this variant 2 has changes in this transactivation domain, and it's really has a different property of induction of HSF driven process. Then what is so everything specific in our amazing niche. So that's why the basic technique is not really applicable. So just this X-MIJ genome has unusual high 80%, it's kind of 80% of 80, it's a quite 80 rich, so you should think about it when you searching this protein motif to activate. It has a lot of trehalose been accumulated to survive without water. And it's this very specific relation to DNA damage, because every desiccation cycle induces a massive DNA breaks during each cycle of desiccation and rehydration. And this genome literally just joined back after every cycle of this drying and reviving back. And what we suppose, but not yet finally characterized, it looks like that the reparation system is based on microhomology-mediated end joining, which is kind of main driver for this kind of restoration after massive DNA breaks. And finally, what we know through the genome analysis, in contrast to Drosophila, for example, this amazing niche has at least less than 1% of true transposons. So it's this is quite puzzling for us. So how it's evolving this kind of evolutionary novelty. Yes, but on the other hand, working with a quite strange creature sometimes gives you quite interesting benefits. Since we cannot use a classical Drosophila and insect promoters, we try, so we get Drosophila, get DH promoter and the IE2 promoters, and it's completely failed to express it into our cells. We had to find some specific promoter, which is found in this amazing niche, and we found one with a quite high expression, and we called it 1-to-1 promoter. Surprisingly, this 1-to-1 promoter, in contrast to the classical one, do not have any activity in mammalian cells. So, driven by our need for our experiment, we suddenly discovered the new promoter that in many cases, much more effective with the classical one and other insect cell line. So this means, apart from desiccation resistance, it's provide us quite interesting insights and give us very interesting biotechnological tool. So this was actually done under collaboration supported by Russian governmental industrial program. Yes, so we try to do now, so we have a very good promoter with a Cas9, and we decided to apply, to make a stable cell line with expression Cas9. And again, the first amazing and striking shock for us was that PB11 cells actually derived from the embryo, and it looks like they're quite sensitive, and just basic expression of Cas9 in the cells been completely toxic, and it's completely devastated the majority of the cells. And here, again, the kind of insights from the biology of this insect plate. What we did, we just added the trehalose. We induced the absorption of the trehalose, how it happens in nature. And interestingly, if you're buffering the expression of the Cas9 by the trehalose addition to the media, so the cells start to accumulate trehalose, and the toxicity of Cas9 drastically drops. So we've been able to compensate toxicity employing trehalose, what we observe in nature, as a kind of a chaperone to stabilize expression. So what we did, so we tried to make a co-expression we have a stable cell line expression GFP, and as a proof of concept, we co-express it with the anti-GFP guide RNA, and yes, a substantial part of cells lost its activity for fluorescence, and it gives us a kind of understanding that it works pretty well. And even depending on the set of guide RNA we select, we can more or less control the exact sites where DNA is actually cut. So we kind of got very basic technology using kind of trick deriving from heterobiopsis. And the second, I'm coming back to this Hitchcock factor story. We've been interested in trying to analyze how this specific Hitchcock factor isoform can contribute to the desiccation resistance. So we took the normal PB11 cells and we employed with a 3-partner system with addition with donor vector. And the logic was very simple, and I believe it's quite elegant. We designed specifically guide RNA specific for the exons we've been interested in, so to kill specifically the isoform of Hitchcock factor, and we integrated under our specific promoter, the GFP. So once we edit the genome, we have cells that do not activate our isoform of interest, but at the same time, it expresses GFP. So the selection can be done by the cells that actually give a fluorescence. And we've been quite successful with that. So the insertion is pretty large, and we can see with the number of clones, it's really acting and it's really working. And after sequencing confirmation, we truly see that cells we selected really have damaged exon of Hitchcock factor in the place we wanted, and it also expressed GFP, so we make it quite elegant and simple selection. And yes, as a proof of concept, we see that although under normal stage, those modified and genome-edited cell line looks quite normal, once we edit it and we start to desiccate, it's practically completely lost ability to survive without the desiccation. So we developed technology and we proved, and from now I hope we will be using it actively for the different topics. And I should confess that I modified my presentation last night after listening all these kind of talks and discussion. And coming to the ability to the edit genome give us potential to do it massively to target different genes of interest. And the question we face now, how to select a proper site in the genome to avoid offset effects or to avoid a side effect. And in our case, it's obvious. So we need to know which parts in the genome are actually contribute to the biology. And this is a question of expanding our knowledge of the parts in the genome that actually contribute to the life of the cell. And for that, I want to make a kind of announcement and to outline another our activity that we are actively invite researchers from Russia and abroad to join us in this thing. So, last maybe 10 minutes or 15 minutes of my talk. Yes, okay, just I have time. Yes, okay. Yes, I will be talking about this. So actually, the same problem what we have is analyzing the non-coding part of the genome is actually overlap with our other projects related to mammalians. And you know, biology of genome consists of the 2 parts. So the first, you always want to know what is a kind of phenotype-specific variants or the risk factor. That's why you analyze DNA. You're looking for mutation. It can be causative, the hereditary type. And when you look on the RNA, it gives you kind of a hint where you look and where can be those parts in the genome where it should be specifically looked at. And you can monitor dynamic of expression. So you know, so when the kind of 1% of the genome are coding genes, but non-coding parts of the genome also actively contribute. And both from our anti-heterobiosis project and our mammalian project, we know that actually every cell in our body has the same DNA, but it's modified and it becomes some specific cell type by activating specific gene program. It's a kind of RNA, which is a driver for cell specialization. So I'm coming from RIKEN, and one technology we actively use based on analyzing not a coding part of the gene, but a non-coding part, which is actually a promoter. So just to remind you, this is just a scheme of a gene, and the actual transcription of the gene can start from several locations, and these locations and the side in front of it cause promoters. And depending on the tissue or some specific activity, the promoters are specific transcription factor bind to the promoters, and actually from a single gene you can have multiple variants of RNA driven by a different transcription factor, and it's actually expand your capacity to see differential expression compared with what you can see with the microarray. So the promoters itself can be a very nice marker for some specific state of cell and pathology. Yes, and we have very interesting and advanced technique. This name is analysis of gene expression, so it's meant to cage. Then actually, in contrast to classical transcriptome, we only select the capped head of RNA. We collect it and we make library that specifically represent 5' ends of the RNA. And we, after mapping it on the genome, we have a very comprehensive mapping of promoters and enhancers across the genome, and we also can see the expression of each given promoter and enhancer. So for example, this is just an example of one gene. Depending on the cell type, the promoter's usage is completely different, and you actually can differentiate it by promoter use. So the Phantom series, driven by RIKEN, been actively accumulating collaborators from around the world, and it's analyzed more than 2,000 different samples that produced knowledge about more than 50,000 of the different promoters, which is actually expanding the knowledge of non-coding regulatory elements by 5-fold from what's been known before. So for example, became apparent that some transcription factor quite specific for the given genes, and it can be one of the kind of driver for the cell specialization. So from a practical point of view, using that analysis, we can actually see that, for example, a fibroblast has a kind of common network component, but if you add a kind of monocyte-specific network and block fibroblast-specific network, you can actually transfer and change the cell type to the different cells. So I remember it was a quite interesting talk today or yesterday about changes of cardiac tissues to the kind of different type of tissues, and probably this is one of the answer and potential way to go. And actually this kind of cell-specific network analysis actually allow you to predict what kind of combination you need to change one cell type to another cell type. And at RIKEN, the specific engine called Mogrify being developed, when you can put starting and finalizing end cell type, and the system will tell you what combination of transcription factor you need to convert the cells. So it's a kind of direct conversion of the cells, excluding IPS stage. Yes, another quite amazing and interesting excitement coming from such kind of project was analysis of enhancers, with a kind of distantly located regulatory elements that also collaborate with the transcription factor, and it can enhance or make a stronger expression of given genes. So Phantom 5 discovered more than 40,000 of new enhancers, and it's been shown that enhancers are really often quite tissue specific, so it's another kind of definition of a cell type by activity of given enhancers. And quite interesting for the medical genetics, that in some of single nucleotide polymorphism in enhancers are actually associated with the given diseases. So technically, don't try to look on these small layers. So it's just a graph representing representation of enhancers, of SNP in enhancers, in promoters, and in a coding region. So far, medical genomics is mostly focused on these coding regions, but technically we found that actually 80% of the causative polymorphism or strongly associated with the diseases polymorphisms are located in enhancers and promoters. So there's a classical dogma of analyzing only coding region, we lose 80% of potential causative region. So it's another kind of very interesting application. Yes, it's just a summarizing. So actually more than 75% of the causative mutations are probably located in enhancers and promoters, which should be kind of focused from now. And Fandom and Trican are now moved to the sixth phase, actively analyzing non-coding RNA. And Russian teams from Novosibirsk and from Moscow are actively participating for this and from a bioinformatics point of view. But in parallel with that, we decided to launch a Russia-specific project that we called Muscle Phantom, where our country act as an organizer, but specifically focus on muscles, skeletal muscles, and how it turned out heart muscles as well. So I will just skip it. And so the interest here is to get more insight about differentiation and the signatures of different muscles in an age-dependent axis in the pathology in normal stage. And it probably can be the first consortium where Russia can be a leader. We already have contributors from 9 countries and 20 institutions, and I actively invite you to join. Just to outline, we have maybe 3 minutes to go. Just to outline, so one excitement. You know, this is more than 600 skeletal muscles in human body. And they come from 3 major origins. This majority comes from mesenchymal progenic cells, and head muscles come from head mesoderm, and extraocular muscles come from precordial wisdom cells. So far, they all called skeletal muscles. They all treated as the same. Phantom 5 has only 2 types of skeletal muscles. But actually, if you remember the destiny of Stephen Hawking, who passed away, he was suffering from amyotrophic lateral sclerosis, but his face been active until very end. And it's known phenomena because it looks like that extraocular muscles that are actually coming from very specific source are quite resistant to the dystrophy in general, including the lateral sclerosis. Why is that? What's made it so special? It's a big question so far, and we hope, as one small piece of our project, it can be very interesting and exciting given. One experiment we do together with the Japanese team is a disuse experiment, as Russia famous for the space science, and we have a project when we will be specifically induce a disuse in one hand of the monkey, and the second hand will be quite normal, and we will see on the level of a single muscle how this disuse, atrophy induced, and what kind of regulatory elements contribute to that and how they act. Does it make sense? Maybe last 2 slides I will kind of talk about that. We have amazingly exciting collaboration with the Dr. Yefimov laboratory from Washington University in the frame of this Russian Scientists Association abroad. And what we tried to do, we analyzed atrium and ventricle from the 16 patients only. We use our approach, we specifically get deeper to that, but only 16 samples, it's just a very few. And what we found, but we immediately identified more than 1,000 new promoters that are specific from the heart. What does it mean for us? These are 100 more locations in the genome where you can see the causative mutation location. The amazing part is we come to the enhancers, but in addition to known 65,000 enhancers, but this very small, very tiny experiment, we've been identifying 3,000 of new enhancers, and only 1,300 been overlapping with our privileged knowledge. Now you can image what we can do if we make 500 different samples from different heart, from different pathology. I think it's going to be quite amazing. So just to finalize, I strongly invite people who work with the muscle-specific cell lines, with the doctors, or everyone who modifies cells coming from muscles or the heart, because together, having one big database and then giving back the results so that you can compare your own samples, which is a big database, it can be quite amazing and Russia-specific consortium-based project. Thank you very much.
Thank you, Oleg. Any questions, please? Yes.
I have two small questions. The first question is concerning this wonderful species which can be dried and rehydrated. So you mentioned that this mechanism for repair DNA is microhomology and joining. Can you comment anything which enzymes probably involved, or do you have an idea to identify this mechanism?
No. I can explain. Yes. Actually, it's another 1 hour talk about this kind of DNA damage relation. So what we know that this image has very specific relation to DNA damage. So you can expose it to 5,000 grays of radiation and you just break DNA into pieces. And it will maintain these pieces of DNA for 2 weeks, slowly assembling it back. And the enzymes that are activating in this kind of irradiated meat or desiccated meat likely resemble those involved in microhomology and joining, and not other types. That's the only insight we have so far. Another kind of insight we have that it has a crazy mechanism, A2AX enzyme that actually phosphorylation of it decides kind of apoptosis and reparation. But in meat, there is no A2AX enzyme. So it's completely lost, trunked with phosphorylation side. So it's a completely different way to survive. So we are in the very beginning of this way, but it's quite exciting for us.
And the second, just practical question, this fantastic database about promoters, I think it would be, probably it's already in internet somewhere, but it would be very important to analyze for the people who does knockout, and this is especially in previous talks what was shown. People usually try to knock out first exon or whatever, first promoter exon region, and this is a mistake because there are alternative promoters, alternative splice variants, etc. And the best way just to use the middle conservative exons for the non-mediated RNA decay.
Exactly. So yes, Phantom 5 is open to everyone, but the only limitation is mostly use primary cells and not so many tissues, so that's why we need to go kind of deeper. But it's available, and actually people use it for that specific things. So in the breast cancer, causative mutation, and so on and so on. So yes, you're totally right. So it's the direction you just mentioned.
Thank you.
Other questions? Yes, please.
Thank you for a very interesting talk. I was very surprised about your results, and maybe are there any links between this number of transposon elements in this insect, let's say, and the stress? Because we know, for example, from plants and from other organisms that any kind of stress that really induce of expression of the transposon is a very important mechanism for evolution and for adaptation. And in this case, I would say that if we would make these the same stress, for example, for plants, we would really see high expression of transposons. But in this case, it's a normal, like a style of life for these animals. How they, how they work with the transposons, how they silence them.
The simple, honest answer, I have no idea. Yeah, so the thing is, since we initially believed it's kind of Drosophila stuff, so it should be like same, it's quite easy to compare. And we found that even compared with a closely related meat from the same genus, the number of transposable elements is really kind of zero. So what we know, we don't have this classical transposons element, but we have very specific, and we not yet know what function is that, multi-copy, extremely conservative, non-coding RNA. And it looks like, this RNA non-coding in its kind of range from 1,000 base pair to smaller one with a really 100% of conservation. It was kind of breaks for our genome assembly as well. So another function of the transposon is giving kind of evolutionary novelty, it just kind of changes. But with meat, naturally exposed to complete DNA damage and joining every cycle of desiccation. So our very naive idea that they don't need a transposon to give evolutionary novelty because they're naturally facing this piecing of DNA and joining it back that actually can introduce more and more innovations. And this kind of natural selection, it's a quite extreme life, and if you mistake, you're completely out, right? But if you have something useful, it can completely be fixed in the population. And we believe, it's quite naively, we don't have any proof, that probably this is kind of one of a crazy way of evolution decided for this pure and loosey insect.
Thank you.
Okay, so I think we have to close the discussion. Thank you again. And before